Okay, hello, good morning. Let me just try and get that up. Okay, is that all right? It's an absolute pleasure to be here. Thank you so much for the invitation. It's nearly 2 decades, actually to the day, that I left CEREGE after what I can only describe as a blissful summer of lavender-scented atmospheres, cicadas kind of chirping in the background. But most of all, I would say the opportunity to interact with some of the most spectacular scientists. And I think that is what CEREGE, aside from all the fantastic science that it's doing, but it's courtesy of a real kind of hot pot of brilliance. And so I will say happy birthday. As mentioned, I work on Coccolithophores. I hope to tell you in this talk why they're so important, although I noticed that Laurent did not include them in his models. They're clearly not important enough for that, but they are to me. And I will start it with this figure on the first slide, and this was actually a figure of data that I was pondering over with Edouard those 2 decades ago. And what I was looking at was the isotopic signals in 4 AMs in the blue, and this is what Edouard was providing, showing these nice glacial interglacial cycles from this subtropical core in the Southern Ocean in oxygen isotopes. And then I'd got a record of oxygen and carbon isotopes in Coccolithophore calcite from the same core. And the challenge was that the 2 records didn't tell us the same thing. So you can see in the top slide that the glacial interglacial cycles in the 4 AMs are kind of fairly regular, but in the coccoliths we had this sort of weird trend that showed, certainly here about 300, 400,000 years ago, very much heavier coccolith isotopic values than the 4 AMs. And then if we look in the carbon isotopes, that's when all hell breaks loose. And you can see, yes, they sort of match here, but when we go back here, the coccoliths are way heavier in terms of their isotopic signals. Now, this may seem a little bit niche, perhaps, for a vision talk, and I hope to show you why I think that these signals are quite important. And I guess that perhaps resonates a philosophy of the way that I've tried to do science, which is proxies are fantastic, but we need to understand them in order to use them. And we need to understand the biology of the organism that is generating the proxy and the signal that we have in our records. So that's the sort of first big thing that I want to say. And then perhaps, I guess, to contrast myself with Laurent, he uses fantastic models. And I'm going to advocate that actually, in a way resonated by Clara's question, the geological record is an incredibly powerful tool if we can learn how to read it. And it provides us with fantastic analogies for us to try and understand the modern system. And so I think those are the 2 things that I kind of want to say in my vision. You've already heard a little bit about this in Laurent, so I'm sorry you're being browbeaten about carbon this morning, but really I also want to say that the carbon cycle is something that we absolutely need to understand, and with an urgency. And that urgency is that we're standing on the precipice of man starting to manipulate the environment to try and absorb carbon dioxide, trying to do any sort of experiment in the ocean to say, oh, well, I think this is going to take carbon dioxide out, so let's have a go. And I think that's a slightly dangerous place to be. I think we should really understand the carbon cycle before we start any kind of large-scale trial. And I think that's also shown in Laurent's talk. There are unexpected consequences of many of these manipulations. So this is just showing the kind of need for the fact that at some point, in order to try and limit our warming in the atmosphere, we're going to have to go to what's called net negative emissions, or where we're actually taking carbon back out of the atmosphere. Again, it's a slightly dangerous place to be, because if we say, oh, we know how to take carbon out of the atmosphere, then of course people will say, well, let's just burn all the carbon that we've got available to us. So really, the message is that we need to try and limit our emissions. That's actually the first order thing that we should be trying to do. And then it is useful to try and understand how we might remove carbon and go towards this sort of net zero or net negative greenhouse gas emissions, and that may be key even for the sort of hard-to-abate emissions from things like aviation. So the bit that gets me really nervous is this plethora of methodologies that are proposed for ocean-based carbon dioxide removal. We've heard a bit about sort of growing microalgae in the ocean. We've heard about macroalgae in the ocean, this idea of deep-sea carbon storage, using electrochemistry, or restoring living blue carbon. That's probably okay. I'm going to focus a little bit on ocean alkalinity enhancement, but really the big question is, and I guess it's in a way the same one as Laura's, how has the ocean acted as a carbon sink, and how will it act as a carbon sink into the future? And just a little bit of chemistry for you. This is what happens in terms of dissolving CO2 in the ocean. So when CO2 dissolves in the ocean, it respeciates to generate bicarbonate and the carbonate ion. And the proportion of those species depends on the pH of the ocean. So the ocean is sat at around a pH of 8.2, and hopefully you can see that at 8.2, the majority of carbon is the bicarbonate ion. The next highest ion is the carbonate ion, and it's actually the CO2 which is the lowest availability in the ocean in terms of its aqueous concentration. And of course, it's that part that equilibrates with the atmosphere. So in terms of the chemistry of the ocean, if you want to try and improve the ocean carbon sink, you've got a couple of ways of trying to do that. One is to try and draw down this CO2, and that's really the way that people have tried to embellish the biological carbon pump, which uses carbon dioxide for its photosynthesis and then essentially pulls this green curve downwards, and that would help the carbon draw down atmospheric carbon dioxide. The other way to do it, and one that I'll sort of think about quite a bit today, is to elevate the pH of the ocean. And this is done by adding alkalinity to the ocean, this sort of weird concept that we have of adding essentially net positive charge to the ocean. And that needs to then be balanced by an increase in the carbonate ion of the ocean, and essentially leads to an increased pH. And so hopefully you can see on here that as pH increases, the CO2 aqueous in the ocean would go down. So that's just to give a little bit of chemistry to the way that we might try and manipulate the ocean. But again, I'm going to look back to the geological record, and I think what the community has done over the last couple of decades, probably since I was in Provence, is to really show us how carbon dioxide has changed. We've been incredibly good at documenting the changes in carbon dioxide, and this is now looking over the last 65 million years or so, and giving us the sort of size of variability in atmospheric CO2 on that kind of timescale. So with estimates going up to about 1600 ppm, and then coming down abruptly about 30 million years ago with some wiggles towards the modern day. So we've done a really good job of documenting what's happened, but I would say that actually as a community, we've got very little understanding about what's actually driving this long-term trend in carbon. We've got some ideas about things like the Earth's thermostat that may well have helped to increase weathering and draw down CO2, but the majority of our proxies do not show peak weathering at this time of peak CO2 and peak warmth. So there's something amiss in our understanding of the carbon system, and I would say this is really where we need to go in the next few years. And this is also true for looking on much more recent timescales. So over about the last 800,000 years, we've got this perfect record of the ice core, which absolutely tells us what CO2, and here I've just got methane on this figure, but really CO2 has bobbed up and down naturally over the last 800,000 years, accompanying glacial interglacial cycles. And this is something that we've known for probably over a decade, the size of these CO2 variations, and the fact that they seem to function on what we think is a kind of orbital timescale, the rate at which the insulation is changing in the atmosphere. But I would still argue, and maybe I'm controversial in saying this, but I don't think we really understand the processes that are absolutely driving this CO2 change, that are defining the limits of how high it goes and how low it goes, and pacing the CO2. We don't have an intricate model that allows us to understand how ice sheets and sea level change and CO2 and nutrient availability are actually making the Earth bob up and down. And so I would say this is, sadly to say it, we've known about these fluctuations for a long time, but I think it's still a frontier to try and understand that. And I think it's ever more acute because we are now in a position where we need to start to manipulate the Earth system. Okay, we finally get to the Coccolithophores and why I care. So these are these fantastic little organisms. They're photosynthetic phytoplankton, and the unique feature of them is that they generate these fabulously intricate calcium carbonate platelets to generate what we call the coccolithophore, the coccolithosphere, or whatever. I'm showing here two different species of coccolithophore. So this is Coccolithus pelagicus, and this is Emiliani huxleyi. And I just want you to notice that they're quite different. So this one is very big, it's very heavily calcified, and what we term it is a high pic to POC. So it's got high particulate inorganic calcite to its squidgy organic carbon cell. By contrast, when you look at Emiliani huxleyi, this is the coccolith that dominates the modern ocean. We capture it in satellite images of the ocean today. You can see the sort of chalkiness of surface waters, but it's really a tiny by comparison to this guy. And not only that, but the list that it generates, and maybe I should have had a bigger picture of this to show, is that they're quite not fully calcified. You can almost see through the liths in contrast to the way this guy calcifies. So that's what they are. Why are they important? They're important because they're actually one of the main, if not about 80% of the long-term carbon sink of carbon dioxide from the atmosphere. So they emerged back in the Triassic. They started to calcify, and they've dominated. And of course, we know them from the Cretaceous, which literally sort of tells us this chalk-bearing period. And they're still persisting and being abundant in the modern ocean. Now, in terms of this Earth's long-term thermostat, this is what I was trying to mention earlier. We generally think about this process of weathering as the main way by which we remove carbon dioxide from the atmosphere. So this is just a very poor, really, geochemical representation of what continental rocks look like, but they react with CO2, and that generates this calcium carbonate of these guys in the ocean, the silica of the diatoms that are also very important. And then, of course, because these things are now photosynthetic, this is only really since this kind of 200 or so million years ago, these minerals are now tied almost also to the formation of organic carbon. And that means they're tied to photosynthesis. And that may help us to understand how this long-term carbon sink has changed in the past. This is just to show you, I was told to make a PDF, so I couldn't show you a really nice video of how these guys generate their calcium carbonate. But this is a little sort of sequence of showing you the process by which these guys make their calcium carbonate. And I guess the point to notice is, look at this, the calcite lith is generated totally intracellularly, and it grows to hit essentially the edge of the cell, and then it pops out onto the surface of the cell. It's the most extraordinary thing to see, that you can see a huge volume of the cell gets taken up, making this calcium carbonate lith. But what that means is, it's all about how the cell responds to carbon in terms of what will dominate and control the amount of calcium carbonate that they make. So a little detour into photosynthesis just for a second. And this is a very important process, of course, for how much carbon gets taken into this biomass, and we've heard a little bit about that. But it's mediated by this enzyme ribisco. And once ribisco was found in evolution, it seems to have really kind of hung around. It hasn't necessarily got a lot better in its ability to fix carbon over time, although the environment has arguably got a little bit more challenging for ribisco to fix carbon. It's a notoriously inefficient enzyme. It binds to carbon dioxide and it fixes carbon, but it's not very good at discriminating between carbon dioxide and oxygen. So Rubisco can also catalyze the fixation of oxygen, which then essentially burns some of the energy that was fixed by carbon fixation. So there's this competitive inhibition between oxygen and CO2, and you can hopefully see that that might make life more challenging now that we live in a highly oxygenated planet. This was great way back sort of billions of years ago, but it's got more challenging for this Rubisco enzyme. And a sort of very, again, a fertile area of research is trying to work out how organisms have tried to overcome the inefficiencies of this Rubisco. I should also say that it tends to have a low affinity for carbon dioxide, its substrate. So there are fantastic models of how we think that organisms have evolved to be able to get enough carbon dioxide inside their cells to catalyze that carbon fixation when living in an oxygenated planet. And these involve things like sort of proteinaceous bodies and packaging of the Rubisco. And most importantly, there's this sort of view of, okay, well, maybe they don't just rely on CO2, but perhaps they use bicarbonate. If you remember, there's loads of bicarbonate in the ocean. There's tiny amounts of CO2. So perhaps if they can turn CO2 or tap into that bicarbonate and get that charged molecule into the cell, they can generate CO2 where they want it and close to the Rubisco. What I should say, though, is that CO2 is an uncharged molecule. And what that means is that it can pass very freely across biological membranes. It just diffuses in and out depending on a concentration gradient, and that might be important for some of the data that I'll show you later. By contrast, bicarbonate is a charged molecule, and therefore a cell has to want to get a hold of that bicarbonate and use some kind of transporter to take it into the cell, and that may add an additional energy component to that cell's growth. Now, whether coccoliths have a CCM, or a carbon concentrating mechanism, or not, is still under debate. This was a beautiful piece of data that Bjorn Rost got many, many years ago now, 2004. And what they're showing is the relationship of your photosynthesis to the amount of CO2 aqueous that you have in your solution. I should say in the modern day, the oceans are poised at around 10 to 20 micromoles of CO2 dissolved in the surface ocean. And what I want to show, and here we're looking at Meilland huxleyi, that small coccolithophore, and then we've got a couple of different diatoms and a Phaeocystis species, I think. And what you can see is that when you increase CO2, those diatoms at modern day levels of CO2 are pretty much functioning at about 100% of their maximum photosynthesis. And that implies to us that those have got a really, really good CCM. They're able to concentrate carbon and accelerate their photosynthesis as quickly as they can. By contrast, Meilland huxleyi is only sitting at around about 40% of its photosynthesis, and as we increase CO2, it accelerates its photosynthesis. So it may have a CCM, but at least what's clear from this is that it still benefits in terms of its productivity from an increase in CO2. And so its CCM perhaps is not as efficient as these guys. So this tells us a little bit about the photosynthesis of the Coccolithophores. But is it possible that they haven't got a CCM? And if not, why not? And this was just a little paper that I was putting together, and I guess there's been a lot of interest in the CCM world in terms of carbon dioxide concentrations. But what I hope I showed you is that Rubisco cares about oxygen relative to carbon dioxide. And when we actually calculate the oxygen to CO2 ratios of the ocean, these range from about 20. You've only got about 20 times as much oxygen to CO2 when you account for the solubility in the surface of the ocean. In the atmosphere, it's a very different world. We've got over 500 times more oxygen than carbon. And so in the atmosphere and for land plants, there's a huge drive for them to generate a CCM. And indeed, this is what we think we see emerging in things like C4 plants and in CAM plants, is this ability to capture carbon. But interestingly, in the ocean, the drive for having a CCM is actually not as great as the atmosphere. And not only that, it probably only started about 400 million years or so ago. What I'm actually showing you here is this equilibrium dissolved O2 relative to CO2 through time, and this is now going over 600 million years or so. And it's just to show that really we kind of only went above oxygen being more abundant than carbon in the ocean, dissolved in the ocean, about 400 million years ago or so. And it's only bobbed up to about maximum of about 40, bobbed down to about 20, and somewhere around in the 20 to 30 mark today. So I'm just trying to say that the driver for a CCM in marine phytoplankton is not as great as it is in the atmosphere. So let's sort of turn to a little bit of peering inside of Coccolithophores. And this work was sort of generated, partly there's been a huge interest in what the ability of these organisms to continue to calcify in an increasingly acidified ocean. And we've been trying to understand why they change their calcification in response to acidification. But more recently, we've also been interested in their response to changing pH, more broadly, where we look going into higher pH world with a view to trying to understand how they may calcify should we start to manipulate the pH of the ocean through enhanced alkalinity or enhanced weathering. And I find this is an SEM that one of my PhD students generated of that Coccolithus pelagicus strain, where we grew the species under low pH, high CO2, and high pH, low CO2. And now we're thinking about calcification itself rather than just photosynthesis. And I guess what I want you to see in this picture is the fact that when we've got high CO2 available, and this is a molecule, as I told you, that can diffuse across membranes, we get these incredibly blocky calcite liths being generated in our cultures. At normal conditions, we get very nice liths. But then if we go to low CO2, you can actually see that it looks as though the lith is not fully formed under low CO2 conditions. And to me, what this is telling me is I can actually see the CO2 diffusing into the calcifying vesicle. And this is quite heretical for those that know about Coccolithophores. We tend to think that they use bicarbonate. Why wouldn't you? There's loads of it. But what to me this is showing is that actually the Coccolithophores are using as their substrate for calcification, they're relying on a diffusive supply of CO2 into that vesicle. Now, in this large species, it also looks as though, and I'm going to sort of say this word, the residence time, the size of that internal pool where they're calcifying is large, and the residence time is long enough that the CO2 can turn into either excess or not enough calcium carbonate. When we do these same experiments with the Meilland huxley, you can see that it's kind of perfectly formed across a whole range of different pHs. This may be that it's using the bicarbonate ion and it's somehow not reacting to the CO2. But as I'll show you, I think its internal pool for carbon turns over incredibly quickly. It's generating these liths at an incredible rate such that carbon almost can't accumulate inside that internal vesicle. Okay, so I can make that sort of conclusion visually, but can I do any better than that? And I'm going to come back to these stable isotopes. So when you dissolve carbon dioxide in water, I've told you generate CO2 aqueous, bicarbonate, and carbonate ion. And these different molecules containing carbon have very different isotopic signatures. So here, this is now looking at the isotopic fractionation of oxygen. This is CO2 aqueous or carbonic acid. They're kind of the same thing. But carbon dioxide has got a very isotopically heavy signal in oxygen isotopes, and it then gets lighter as we go towards the carbonate ion. And this indeed has been attributed or shown to perhaps play a role in foraminifera. By contrast, if we look at C13, there you can see that the CO2 molecule, at least at pH 8, is always about 10 per mil lighter than the carbonate ion and the bicarbonate ion, or 9 per mil than the carbonate and 10 from the bicarbonate ion. So CO2 is always very isotopically light by about 10 per mil. remember that number. Oops. Gosh, I hope that wasn't me. Okay. So what do isotopes tell us about the sources of carbon for calcification in coccolithophores? Now, it's taken me a long time to work this out. We did some cultures back in 2010 where we grew the heavily calcifying and the smaller coccolithophore. And we were able to show that the isotopic values of the calcite seem to depend on the availability of CO2. So the large coccolithophore had this isotopic fractionation down to very light isotopes when I'm growing them at low CO2. By contrast, the smaller coccolithophore was kind of the same across these CO2 gradient. We followed this up with some further culture experiments with Harry McClelland in 2007. And again, we found that this is now looking at the isotopic offset of the coccoliths relative to the carbon that's in the culture. And I'll just draw your eye. So in the top graph, we're looking at the calcite that's generated. And in the red, we're looking at a small, low-pick POC species. And in the blue, we're looking at a heavily calcified species. And we can see this splaying of the vital effects at low CO2, which sort of resonates with what we found earlier. And in this paper, we attribute this to the degree of calcification of the cell, and that the heavier, heavily calcified coccoliths kind of trend towards the source of carbon as they're growing, whereas these guys, which are photosynthesizing an awful lot, remove light isotopic carbon and leave the carbon isotopically heavy. Now, the challenge in coccolith world is that everything is sort of correlated, actually, between these species. So if you're heavily calcified and a large cell, it seems that you also grow slowly. If you're small, you tend to be lightly calcified and you grow very quickly. And so it's quite hard to entangle which of these things is really driving this change. What Nick Chauhan did, another graduate student who's now in Cambridge, he also grew the coccolithophores in a range of CO2 by manipulating the pH. And we can see that there's a very different optima in terms of CO2 and in terms of pH for these heavily calcified species. They tend to grow the fastest at a value of about 20 micromoles per kilogram. They drop off at higher CO2. Everything drops off as we go towards very low pH. It looks as though there is some pH inhibition. But as we go to lower CO2, what you can see is that Meilland huxleyi grows really fast. It's very good at living at very low CO2. And not only that, but it grows much faster than even the optimal growth rate of this Coccolithus pelagicus. So even from this growth curve, we might make the argument that Meilland huxleyi is really good at getting carbon for its calcification, whereas Coccolithus pelagicus is somewhat more limited by low CO2. Let's now look at the isotopes, and this is a horribly complicated plot, which I hope I'm going to get through, and I'm now realizing that I'm going far too slowly. The main thing to pick up, this is now looking at the carbon isotopes in the coccoliths relative to the oxygen isotopes in the coccoliths. What I want you to pick up is that the big coccoliths, when they're CO2 limited, tend towards values of about -10 per mil of carbon in their calcium carbonate. So this is a value which is very similar to that of CO2. And this sort of starts to build this picture that they're relying on diffusive CO2 for their calcium carbonate. The way that we preserve that CO2 signal inside the coccolith is it must have a sort of depleted internal pool under very low CO2. And that means that all of the carbon that's coming into the lith is quantitatively transformed into the calcium carbonate. By contrast, if we look at the small cells, which are growing very quickly, at low CO2, what you can see is their isotopes go very isotopically heavy in both carbon and in oxygen isotopes. They seem to fall along a gradient with a ratio of about 1/2. And that's what we would expect for mass-dependent fractionation, and that is often attributed to diffusion. So here we're seeing some impact of CO2 diffusion, but these are going isotopically heavier than their source carbon. So what that implies is that actually they've got to have concentrated carbon within the cells for the calcium carbonate, and that what we're seeing is CO2 diffusion out of the cell. So it tells us that Meilland Huxley is actually concentrating carbon in order to be able to calcify, and most likely is using bicarbonate transporters to help its calcification. But by contrast, this large guy is still reliant on CO2 for its calcification. I think I won't go through the majority of this. And so we have these very different uses of ions for calcification in these contrasting species. So what does this mean, or can we take some of those ideas into experiments where now we've actually raised the alkalinity in the solution? In Nick's experiments, we were just manipulating pH, but here we're actually raising alkalinity in a way to try and understand calcification in response to what we might do to the ocean. And what I want you to see sort of almost immediately from these cultures of Meilland Huxley, and we've done this for Coccolithophores braurudii, is I hope you can see that these are kind of denser than these cultures. And this is where we've raised the alkalinity, so modern alkalinity about 2,500, and we've gone up very extremely, I would say. This is way beyond anything that we're talking about in terms of ocean manipulation. But I hope you can see that Meilland Huxley seems to grow incredibly well under high alkalinity. And this was borne out in the growth curves. We're looking here at the cells against the days in these elevated alkalinity experiments, and you can see the control here. It grows, and we get less biomass at the end of the experiment, and its growth rate is lower than those that we have growing in the higher alkalinity conditions. And this is just showing the sort of integrated growth rate for that. So we've got low growth rate of Meilland Huxley. It's somewhat limited under modern ocean conditions, and then it accelerates when we go to higher alkalinity. Now, I'll try and sort of whiz through this rather quickly, but what we seem to see, so in the top graph, what we're looking at is the growth rate versus a few of these carbonate parameters, which are all essentially correlating through these experiments. And in the blue, we're looking at G. Huxleyi, and in the red, we're looking at Coccolithophores braurudii. And I guess what I want you to notice is when we raise the alkalinity, we do what we expect. We equilibrate these with the atmosphere, which means that we raise the bicarbonate ion in those experiments, and we see an increase in growth rate in both species associated with these experiments. So overall, the alkalinity seems to be fertilizing the growth of these organisms. What's interesting is that when we look at the calcification rate, though, what we actually see is the calcification rate is decreasing with this elevated alkalinity in both bicarbonate and in total alkalinity. And this is sort of curious. So it sort of firstly, it helps us say, okay, well, bicarbonate, at least in these experiments, is not the driving substrate for calcification, because we're seeing declines as we're increasing what we could have as the substrate. Instead, what we think is actually happening here is that the growth rate is going so fast, or has accelerated so much, that the CO2 diffusion for the calcification is not keeping pace. And it means that per cell, we're reducing the amount of calcite. And we see this, at least in morphological changes, in the big guys when we grow them under the very high alkalinities. They're declining in their calcification rate, and we see morphological problems which could be related to CO2 limitation. So whilst the raising of alkalinity will fertilize the growth of these organisms, the calcification may well decline in each of these species associated with CO2 limitation. And I should say that we think that the Meilland Huxley is only upregulating that bicarbonate transporter when it's growing under CO2 limitation. And we very carefully manipulated these experiments such that there was no CO2 limitation of the growth rate. So it seems that when CO2 is not limiting the growth rate, their calcification is reliant on CO2 diffusion, and it declines as the growth rate goes up. Now, is this just a culture curiosity, or what is this? And this is some beautiful work by Alba Gonzalez Lanches, who has been looking at the degree of calcification from sediments in a core top transect across the Atlantic. So going from sort of 0 up to 70 degrees north and down a little bit into the south. And I don't have time to go through all of this, but she's basically counted the coccoliths per gram in the sediments. And we see that the number of coccoliths in the sediments seem to represent a species optima in the environment. And so we see these kind of big species optima here for Coccolithus pelagicus in the environment, and for another large, heavily calcifying species, their optima seems to be hitting further north than about 40 degrees in the ocean. But what's interesting is when we look at this size normalized thickness, which is a measure of the calcification intensity, it's very much decreased when they're growing really well. So even in the modern ocean, it would seem that when they're able to grow very quickly, the CO2 diffusion is unable to keep pace with the large demand of carbon for the calcification, such that the calcite gets actually thinner in their production. By contrast, if we look at the large Gepharocapsids, the small Gepharocapsids, we actually see that the thickness goes hand in hand with their optima. And because they're smaller, they've got a lower demand for carbon, it would seem in the environment that calcification can keep pace with that optimal growth. But what's nice about this is it finally makes sense of some data we got years ago looking at the average coccolith mass over the anthropogenic timescale, and this was looking in exceptionally accumulating sediments in the North Atlantic, where we got this kick up in the coccolith mass in the very recent anthropogenic times, which we were attributing to a CO2 sort of fertilization of calcification, but it was very hypothetical at the time. And Paul Halloran, a graduate student who was leading some of this data, was able to look at the sizes on a Coulter counter. And essentially what we were able to show is that what this was being driven by was really a trend towards greater Coccolithus pelagicus. And so as CO2 has gone up in the environment, it has accelerated the ability of Coccolithus pelagicus to calcify and perhaps become more dominant in the environment. And we're seeing this in the sediments. So this sort of little mechanistic understanding is really helping us understand the adaptation of the biogeochemistry. And this plays out in the geological record. So this is a current graduate student, Tom Wood, who's trying to look at carbonate production. And now over the last 70 million years, I apologize for the grimness of this slide. For some reason, I could not find strontium isotopes as a database, but there we are. So the key thing to note here is this is Tom's estimate of calcium carbonate productivity. So we're thinking about vertical accumulation rate of calcium carbonate. We get this sort of depression way back in the Eocene and Paleogene, and it really kicks up about 40 million years ago, and this is totally coincident with the kick up in the strontium isotopes, which may be reflecting something to do with the weathering input. So I guess my message for those that are thinking about ocean alkalinity enhancement is, if we put more alkalinity into the ocean, the coccoliths, I'm afraid, are going to suck it up in their calcification alongside the carbon. And so I don't think that there will be an increased reservoir of alkalinity to absorb the CO2. I suspect the coccoliths will take up that alkalinity for their calcification and sink it to the bottom, which still acts as a carbon sink, but not the way that those that are trying to think about it think. Okay, I'm just going to come back to a final slide, and this was to sort of circle back to the data that I had with Edouard all those years ago, where I showed you right on the first slide the sort of deviation of the coccolith isotopes from the forams, and what on earth did that mean? I've now plotted on this horrible graph, I'm sorry, you've got to look at this again. I've plotted on this horrible graph the carbon versus the oxygen isotopes for that core, MD962077. And what I hope you can see is that by and large, the gradient of that data seems to follow the gradient that we were finding in our culture coccoliths grown at different CO2s or different pHs. If you remember, there was that period of time about 400,000 years ago where the carbon isotopes went really heavy in the Coccolithophores. And what I think is we can actually interpret this now So the gradient looks like that in the gyrocarpsids, but what that heavy isotopic signal that we had is accompanied by an acme of heavily calcifying coccolithophores at the time, where they're growing very quickly. So what we're seeing in their isotopic signals is actually CO2 diffusion out of the cells, peak around 400,000 years ago. Now, what's driving that CO2 diffusion out of the cells? It could be environmental. So it could be driven by low external CO2, but we have the ice core records that perhaps say, okay, there's not really exceptionally low CO2 at that time. Or it could be driven by high external alkalinity, which is drawing CO2 out of those cells. Or it could be that they're growing very quickly and therefore need to pump loads of bicarbonate, and therefore that's leading to a greater diffusion of CO2 out of the cells. But it was just to show that we're finally getting close to understanding that data. Okay, I've gone on for a very long time. My conclusions are that I guess that the main takeaway is to say that if we want to work out how we're going to store carbon, I really think we need to understand the past and the broader impacts of different scenarios. And then I've just tried to show you a sort of vignette of how understanding coccolithophores can tell us more about their response to carbonate chemistry, but also how they may have played a role in the carbon sink in the past. So thank you for your attention.
Thank you so much, Rosalind, for this really interesting talk. Some questions?
Thanks, Rob. It's super interesting to see. Oh, sorry. No, sorry. Super interesting to see the difference between those big and small species. And I guess I have one question, which, so how do you think that relates to kind of the alkanone producers and their ability to tell us about, you know, potential past environments? Because I've always thought of it as this kind of potentially one-way track of evolution towards producing a coccolith that loves low CO2 environments, like G. hux, which we now know produces most of the alkanones that we see in the sediments. But how does that relate to the kind of past and future of the alkanones as a proxy for the carbon cycle?
Okay, great question, Tom. I would refer you to my Goldschmidt talk, but so the bit that I didn't talk about, and God, this could be a really long answer. The short answer is we've had a trend towards smaller, faster-growing species. And I think the trend, and that's in the reticulofenestrids coming into the gyrophycapsids. I think the alkanone record that we see is, what we're seeing in that record is a measure of the assemblage of the alkanones that are generated by an assemblage of species. I might circle back to why I say that in a second. But what I think has happened is the cells have undoubtedly got smaller and less calcified as CO2 has changed in the environment, decreased in the environment. But what I think the reason why they've got smaller is almost to try and make their calcification or their production keep pace with their carbon supply. So what we find actually no, this is not the graph. Okay, so what we find in the carbon isotopes of the liths tells us also about the residence time of carbon in the cells. And the gyrophycapsids have got a very low residence time of carbon in the cells, whereas the large guys have got a high residence time of carbon in the cells. So why that's important is essentially, I think they've got smaller to keep the reservoir full. I don't think in the smallest cells that there is sufficient time for Rayleigh fractionation to happen. We see Rayleigh fractionation in the larger cells that are slower growing because the carbon's had time to Rayleigh fractionate. So what I think we're seeing in the alkanone records, and this is horribly controversial, but we did have, in Nick's paper, we did look at the carbon isotopes in the organics that were generated by the cells. And you kind of see this relationship almost between how sensitive the organics are to CO2 and how sensitive the isotopes are to CO2. So this is what we're sort of showing here, this thing, which tells us about the carbon residence time. So what I think has happened is we actually know the fractionation factor for E-Hux's Rubisco is 11 per mil. So that's much smaller than the 25 per mil that's classically used as spinach. If you look at the alkanones today, they're about 18 per mil offset from the ocean as a sort of bulk-ish kind of measure. So 18 is not 11, and that's why I say that it's reflecting this assemblage change. And what I think has happened is they've evolved, keeping the internal carbon pool full, which has allowed the Rubisco to relax, and that's why you get an 11 per mil isotopic fractionation in E-Hux as opposed to 25 per mil in something else. Now, we don't have the Rubisco fractionation factors for a range of different coccolithophores, sadly. But my overwhelming view is that what we're seeing is sort of almost the impact of this internal carbon residence time that's allowed Rubisco to relax. We've got a little bit of data where we've looked at sort of positive selection, and we're seeing that relaxation happening sometime in the last 100 million years. Downside of genetics is it doesn't give you timing. And so that's my view of alkanones. I think they're really telling us about the assemblage. It's an assemblage average of their Rubisco fractionation factor. Sorry, but which is still sensitive to CO2, right? But actually, I think the cell size is almost more important, and the degree of calcification.
Rose, yes, thanks for the wonderful talk. Back to the modeling, maybe. So we do have some calcification implicitly, so we do represent coccolithophores implicitly in the models, but we don't have any influence of pH, CO2 on the calcification rate, because I don't know what to do. And so your recommendation would be, if you want to do it, you need to have two types, highly calcifying, or the one that don't calcify so much, because that's the only way to represent what you show here today, no?
I think that's absolutely right. I think, and this is something that Jorentje and Hendricks and I talked about many moons ago, but is the bit that is not really recognized sort of more broadly is that, in fact, the big guys, the coccolithophores, branched off from the isochrysidales kind of tens of millions of years ago. Somebody can maybe give me the date because I can't quite remember, but and not only that, but the isochrysidales have largely either lost and regained calcification or some combination of this. So the point is that Meilland huxley is a very unusual coccolithophore compared to Coccolithus pelagicus and the majority of the coccolithophores. And the two groups are very different. It seems Meilland huxley and the Japhairocapsids are all about growing fast. They're sort of, I'll get it wrong, the K strategist, the R strategist, but they're all about going fast. And the big guys are all about sort of hanging out and turning over much slower, but therefore being much more big and much more massive. And so, yes, I think that therefore they have different ways of handling carbon. They may have different ways of handling phosphate. We know that E-Hux can get hold of alkaline phosphatase. It uses alkaline phosphatase to get hold of organic phosphorus. The big guys, we don't know. But our best look at their genome suggests they don't have an alkaline phosphatase. So yes, I think they're very different and having two different and what we also see. Whoops, I whistled over it. I'm so sorry. I sort of whistled over a lot of detail. But the coccolithophores are kind of dominating in the high latitude, so sort of further north than about 40 degrees north. They seem to pop up. They pop up in upwelling areas, and they do exist in the Southern Ocean, but they're quite marginalized, whereas E-Hux is sort of dominating in that kind of subtropical, lower tropical realm. So yes, they're very different.
Rose, very good talk, thank you. I was surprised by your conclusion on what you've shown from the first graph. And just before the conclusion,
this one.
yes, you mentioned that you maybe find the reason why you had, when you were in CEREGE, found the delta C13 on a very strange one for the Coccolithophores, but the delta C13 of Coccolithophores is going for 4 climatic cycles, the Geophyrochapsa bloom, which it lay for 400,000 years. And the CO2 in that time went to 4 climatic cycles. So they had changed a lot. And alkalinity, I don't know, but I don't know how you can explain from that graph the first graph that you showed. And I'd like to comment on that.
So from this graph, what I can say, I guess what I can say, I was thinking about this because I bunged it together last night, if I'm honest, because I was like, oh, I should sort of try and come back to the end or the beginning. So what I can say is these are sitting at a place where we know that we've got peak CO2 diffusion out of the cell. So that's what I can say. I think what I can understand from this is the species that were dominating 400,000 years ago have peak CO2 out. That's kind of what that signature tells me. Now, why and I thought, I've cracked it. And then I sort of realized, oh, crikey, why have I got peak CO2 going out? And then I realized it was more complicated. Now, I totally agree with you. So yes, you're absolutely right. If I go all the way back to the beginning, that yes, it's a very curious sort of 300,000 or 400,000 year event when I've got peaks, and there are bumps, there are sort of glacial interglacial cycles in it, right? But these bits I've got sort of peak CO2 out of my cells, if you believe me. I think that is what is driving the vital effects in the small guys. So as I say, one possibility which sits with your hypothesis maybe and my hypothesis is that these things are growing really fast. That's the one thing that we sort of know and probably agree on. And indeed, the strontium calcium is sort of telling us that they're pumping calcium at the sort of highest rate, so they've got the highest strontium value in the calcium carbonate. So if they're growing really fast, then they may be pumping bicarbonate really fast. So it may be that this is telling us about the growth rate of these guys, that they're growing fast, pumping bicarbonate, and that's what's giving you the high CO2 diffusion out. Basically, the cell is driving a greater gradient. I guess the only thing that I would say is I think the time scale of this is really curious, the sort of 300, 400,000 years, as you and I have debated a lot. And it's just really interesting to me that that's the sort of timescale for the residence time of alkalinity in the ocean. And so I find it sort of intriguing that the length of this is maybe resonating with the biogeochemical residence time of alkalinity. And you know, with Steve Barker, we did some of the modeling to sort of say, okay, well, what do we need to do to the sort of alkalinity DIC budget? And there is a way of kind of manipulating the carbonate chemistry such that you don't see it in atmospheric CO2. But it's still, I don't know. So I haven't fully cracked it, Luke. I can't believe it. It's nearly 20 years and I still don't understand this record. But I got a little step closer, I think.
One more question before break.
Hi. You mentioned the size, and when we are looking at evolution of Coccolithophores in the recent, I would say, Neogene, we have two events that could be interesting. Sorry, yeah, two events that could be interesting. There is the migration of Coccolithus pelagicus up north at the beginning of the Pliocene, but also the reduction of size of coccoliths, size and mass, during the end of the Miocene. Do you think the main driver could be the CO2? And if yes, could measuring coccolith could be a proxy for CO2 at the end, which is way less expensive than doing boron or whatever you want to do? Thanks.
It's a great question as to what's driving the end of the Miocene. And and why the coccoliths get smaller, I think looking at size fractionated vital effects would be, I mean, all I would say is that doing size fractionated vital effects is kind of boring. It takes a long time to generate those size fractions, and all of, I guess most of the data that I've shown you is actually just done on the bulk coccolith fraction, which has its sort of complexities because it's assemblage dependent. But yes, it would be fantastic to do it. I mean, there's a record that Mikhail Hermoso, a sort of brilliant postdoc in my lab, generated together with a fourth-year student, Sam Hurst, and we have tried to look at the Miocene, and it's slightly, and I guess just as that, but what we found there was that the vital effects seemed to emerge in the 5 to 8 micron fraction, but it was very noisy, and so it was hard to really pull out a CO2 signal. But yes, I think clearly at the end of the Miocene there is something that's changing in the Coccolithophores, and it could be CO2. There may be another driver.
So thank you so much, Rosalind, for the nice talk.